Good evening, everyone, and welcome. We are thrilled that you're joining us tonight, alumni and friends of the Woodsworth College Alumni Association. Even though this is an online event and we're not physically at Woodsworth College, we are here within the realm of the university, and so we would like to begin with the acknowledgement of traditional land. We wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron, Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across the Turtle Island, and we're grateful to have the opportunity to gather and work on this land. For this lecture, everyone, there will be a Q&A session at the end, so please write your questions and comments in the chat, and Professor Khan will address them in the discussion to follow the lecture. Then for the final remarks, our president of the Woodsworth College Alumni Association, Erin Thompson, will have the chance to thank everybody. It's with great pleasure that we are here for the second Alumni Cafe event this year, the first one in 2022. We're delighted to welcome the speaker of tonight's lecture, Professor Omar Khan. Professor Khan is the Canada Research Chair in Nucleic Acid Therapeutics at the Institute of Biomedical Engineering and Department of Immunology here at U of T. Professor Khan is also the scientific founder and former chief scientist of Teva Biotech, a Boston area company spun out from his postdoctoral research at MIT. Currently, Professor Khan contributes to Canada's growing research by creating new regenerative medicine, enabling technologies, and training highly qualified personnel capable of addressing diverse local and global bioengineering needs. As an RNA nanotechnology and vaccine expert, Professor Khan is regularly featured in the news where he shares science in an accessible way. I have come across Professor Khan while he was on one of his media appearances on CTV, and for that we thank him for keeping us and the public informed during these past 2 years of COVID, making sense of the science to make informed decisions. With the growing interest in mRNA technology and where else it could be utilized in medicine, this topic is important and critical for all our well-being. Professor Khan, we are very grateful that you have made the time to join us tonight and to present us with your interesting topic, evolution plus engineering equal nucleic acid nanotechnologies to cure diseases. Without further ado, Professor Khan, the floor is yours, or in a more timely expression, Professor Khan, the screen is yours.
All right, well, thank you everybody for the very kind introduction. I'm excited to get started. All right, so today's talk is going to be a fun one, I feel. Let's start with the beginning. How did evolution get us to where we are now? Well, diseases are complex and they're caused by multiple problems. There are some diseases that can be caused by a single thing that's gone wrong, and perhaps that is addressable by something like a small molecule drug, and you can cure it very easily. But unfortunately, all of the really challenging diseases like cancer and variety of chronic viral infections, they're caused by many things, and the opportunity to address them from a approach that targeted many things at once to try to rally everything together, it would be useful. So one of the things we do is that we start looking at those very complex diseases, the ones that we can't treat with drugs because they're just too hard to do. And we are fortunate because we have been gifted something through evolution that we can potentially use to help us achieve our goals. And those tools are nucleic acids. We have a broad range of different types of nucleic acids. So essentially, nucleic acids are instruction sets. They're instruction sets that your cells can read and follow. So if we can potentially give your cells the right instruction sets, we can help your cells recover, maybe regenerate, and do all sorts of things. So it's a very powerful and fundamental tool. Now, there are many different types of nucleic acids. There are mRNA, and they're used to express a gene. So if you send in an mRNA, and the mRNA gets read by the cells, and the cell makes a protein. We have small interfering RNA, and that's used to silence genes. So if you have maybe a defective gene, and it's being expressed, and the protein that's made from that is causing a disease, we could potentially turn that gene off temporarily with a small interfering RNA. MicroRNA, they're different. They are non-coding, which means they don't turn into proteins. In fact, they bind to things in your cells. And when they bind to things, they change cell behavior. So they have a regulatory role. Of course, there's much talk about gene editing and small All right, so I'm back. Okay, good. So we have single guide nucleic. So we have guide RNA that can help us tell gene editing tools where to go and cut out genes that might be defective or need replacing. And we'll also have replicons. These are self-replicating mRNA. So when you put them inside of a cell, they make more copies of themselves so that they can amplify their effects. So there's a lot of different things. And in general, what my lab does is that we make nanoparticles that carry a variety of different nucleic acids all together. And then we add them to cells. They can do multiple things at once. So rather than doing one thing to cure a disease, we're looking at very complex diseases that have multiple mechanisms of action. And we put in a variety of different nucleic acids that then control multiple events to get us that therapeutic outcome. So let's kind of start off with what got us here in terms of the pandemic. So the pandemic, we have the novel coronavirus that's come up, and we had this opportunity to use mRNA-based vaccines to kind of help us out. But mRNA vaccines aren't new. They've been around for decades. They've been in clinical trials for over a decade. And so not new to us, but new to the general public. And I think that's something that's worth noting because they've been in clinical trials long before what we see with the COVID vaccines. But let me take you through the story of how we kind of got here with mRNA vaccines and what was the drive behind them. Well, to do that, let's talk about infection. So when you're infected, you're sick, lying down in bed, maybe you've been infected by something like a virus or even a bacteria or a parasite. In all cases, those things are using you as raw material. Their only job is to make more of themselves, and they're using you as the substrate to make more of themselves. Now, if you're infected, you have a couple of options. If it's a virus, you can take an antiretroviral. If it's something like a bacteria, you can take an antibiotic. And in these cases, what they do is that they slow down replication. So the pathogen is not able to replicate as efficiently, so their numbers stay low. And if we suppress their ability to make more of themselves, your immune system can have a better chance at fighting them off. And then ultimately, we get two important things. We get our antibodies and we get our T cells. These are two important components of your immune system that help you detect pathogens with the antibody and help you kill infected cells with T cells. And this way you can clear infection. Vaccines have always been a very powerful healthcare tool. They save millions of lives a year, and they're a shortcut. They save you the trauma of going through the infection event, and they get you to your antibodies and T cells, which is phenomenal because who wouldn't want to save trauma? And let's look at how vaccines were originally made. So this is almost actually over about 90 years old now. This is one of the original ways we used to make vaccines. We used fertilized chicken eggs. So we started off with the pathogen. In this case, I'm showing a little virus. So the virus, we want to make a vaccine against this virus. So we used to take the virus and we used to inject them into fertilized chicken eggs. These eggs would have grown into chickens otherwise, but now they are flooded with virus because the virus is using the chicken inside the substrate is making more of itself. And then you crack open the egg, you collect all the liquid that's loaded with virus. And you take that and you can treat it. So you can inactivate it, so you can treat it with heat or some kind of chemical to disable it, and then inject that as the vaccine. And then it's so weakened that your immune system can easily overcome it and make those antibodies and T cells, which you need for protection. You can also just break it up into smaller fragments and then pick a unique part of it. So a part that's uniquely found on the virus and nowhere else. So it's not found in your body, it's just unique to the virus. And then we inject that into the body, and then the immune system will respond to that and develop its antibodies and T cells against that. Now, it works fairly well. It's not great for all types of pathogens. It works really well for viruses, but if you are trying to make a vaccine against bacteria or a parasitic flatworm or anything else like that, it's not really going to work. So it's very specific, and it takes quite some time. By the time you have a purified virus ready to go and you want to make a vaccine from it, it takes 6 to 8 months to produce this. So it's a very long time, especially when you're thinking about responding quickly to pandemics. Now, that's how we were making it for quite some time. And back in 2009, there was another pandemic, not as bad as this one, but it was the H1N1 influenza. And we kind of knew how to make that vaccine, make them with fertilized eggs. But the reason we couldn't get vaccines out in time was that there was a shortage of fertilized chicken eggs. And that was the break point. And unfortunately, that forced people to think of new and better ways to make vaccines. How do you remove the chicken egg part of it and just make it without them? So the idea is that the chicken egg is just basically a bioreactor. You just need some cells in a reactor, and then you add the virus, and the virus will infect the cells, make more of itself, and you collect that. So get rid of the shell, get rid of the egg, and just do it that way in a little bioreactor. And that's exactly what's done. In fact, many of the flu vaccines that are used today are made in these bioreactors, and quite a bit are made all over the world, including in Australia and Europe. Many of the vaccines are made this way now, and it's fantastic. And this is an example of the Novartis pharmaceutical plant. They sold it off to another competitor recently, and they make flu vaccines in this way, and it's great. But the challenge remained that this still takes several months to make. So with that, is there an opportunity to shrink that time even more? And that's where the idea of these nanoparticles come in, because the idea is, rather than using a reactor or a chicken egg to make your vaccine, why don't we send the instructions in to your body so your body can make them? Your body is great at making stuff. It can make all kinds of proteins that built you, and there's an opportunity to take advantage of that and use your cells as the production factory to make the vaccine to keep you healthy. And the idea is that we have a couple of components. We have a delivery molecule and some helper molecules, and you combine that with an RNA that encodes an antigen. And then you form your nanoparticles and you're ready to go. And if you can inject this into a person, that person, their cells will read those instructions, make the antigen that'll engage with the immune system, and then you can develop your protective immunity. The great thing about this is because you'll only really have to make the RNA. And as long as you make the RNA, the rest of these components are the same. So it becomes a platform. So you just have to swap out the RNA, and it's fast. You can do this in as little as 4 weeks. So that's really good for responsiveness. So I'm going to take you through some of the case studies of what, some of the work that we were doing. And this was a bit fun because we were using an exotic type of mRNA, a self-replicating mRNA. And I'll tell you a little bit about it. So I had a good friend, Chris Cooper, who used to work for the US Army Institute for Infectious Disease Research in Fort Detrick in Maryland. And we were working together on making an Ebola vaccine. So this is the Ebola pathogen. And what we did is we picked an antigen. So over here, the Ebola glycoprotein. This is a protein that's unique to Ebola and not found anywhere else in your body. So we took that antigen, we sequenced it, we made the corresponding mRNA, and we connected it to a replicon template. And this is what we ended up making. We made a special type of mRNA. It looks like a regular mRNA, but it has two parts to it. The first part are instructions to make an enzyme that self-replicates. So this enzyme makes more copies of your RNA. And the second half are instructions to make your antigen. So if you can get this inside of your cell, what happens is that your ribosome translates it, it makes the enzyme, and it makes the antigen. That enzyme makes more copies of the RNA, and then your ribosome makes more antigen, and it goes into a cycle. So it self-amplifies, and we can control the kinetics of this. So it's not permanent and only lasts a few weeks, up to a few weeks, and you can tune that length, but it was a great way. The idea was that we wanted to make a vaccine that doesn't need a boost. So it will continue to boost itself for a fixed amount of time, and then you're good. You don't have to go back in for a second or third dose. Now, that's great. So we had the RNA part sorted out. And the next part was, how do we deliver it? So at the time, many people were making vaccines and they always included an adjuvant. So an adjuvant is an irritant, basically. So it alerts the immune system. The immune system sees the irritation, it goes to the site of irritation, and it sees all the antigen and says, okay, well, I guess the irritation is being caused by this antigen. I'm going to develop a really robust immune response. Now, that's great for normal vaccines, but our hypothesis was that that's not great for an mRNA vaccine. And here's why. When a cell is irritated, it locks itself down. The last thing it wants to do is follow mRNA instructions. So our argument was that we want to develop a better delivery system and that is adjuvant free because we don't want to cause any irritation, because the irritation will make a cell lock down and not want to express mRNA, which defeats the whole purpose of giving it mRNA. It won't want to follow our instructions, so let's kind of stop that. So we decided to show an experiment where we used one of the delivery systems that I designed and use them as a vaccine with or without one of the most popular adjuvants at the time. So here's what we did. We had a good working Ebola vaccine that we had made previously. So we took that. The vaccine was what we call multivalent. So it didn't express one antigen. It's sort of like these COVID vaccines. They only express the spike protein. We expressed two antigens, a glycoprotein and VP40. So these are both unique to Ebola. I put them into a single vaccine. Chris vaccinated the animals, and then we saw what happens. So this is a survival curve, and we had control animals, and when you give them Ebola, everybody dies. So what we did next was that we used a low dose of our vaccine. If we use a very high dose of the vaccine, everybody would survive. So we had to use a lower dose to be at the threshold of protection so we can see differences. So what we did is that we used the low dose of our vaccine, and we said that, okay, with the low dose of vaccine, we get 6 out of the 7 animals, they survive exposure to Ebola. That's great. Now, look what happens when you use an adjuvant in this case. When you use an adjuvant, you actually only get 4 of the 7 animals surviving, and that's because the adjuvant is locking down their cells so they don't want to express the mRNA we're giving. So that's kind of our point to show that it's very important to not just think about your mRNA, but you really have to think about how you deliver that mRNA. And that's because those two parts are fundamental to the vaccine. You really got to get both right. So in parallel, separately from us, Novartis Pharmaceuticals, who at the time was working with a similar system, found the same thing. They said that if you adjuvant at the wrong time, like adjuvanting very early, you diminish the efficacy of your vaccines. And this was really great because we're understanding now how to make even better vaccines. So with this in hand, we took this platform and we tried it on a variety of different diseases. So with Zaire Ebola virus, we had 100% protection against Ebola virus. Venezuelan equine encephalitis, this is a very bizarre virus. It was weaponized in the Cold War by the Americans and the Russians. They never used it on each other, but it was developed. And just one virion in a cloud is enough to kill you. It's just crazy. And so they kind of created a problem. So we tried to create a solution for it. And so we made a VEE vaccine, and that was 100% protective. We tried it with influenza, also 100% protective. And we even kind of looked at viruses that cause cancer, so HPV. And the vaccine was fully protective there. So that's great. These are all good for treating, straight viruses. But one of the things that I was really interested in, and still am, is what is affecting the developing world. My family is from the developing world, and it's really important for me to remember that there's a huge part of the population that suffers from very different diseases, things that we don't see in Canada at all, but they deserve protection as well. And one of those things is Toxoplasma gondii. It is a bizarre parasite. It infects 1/3 of the world's population. And that's both in developing world and in developed countries. So, 1/3 of the audience out here probably has it. It can eventually, it can cause cerebral toxoplasmosis. So if you have it and you get really old and you start getting dementia, sometimes it's caused by this little parasite living in your brain. And there's no approved human vaccine. And the reason why is because it's very difficult to treat. It has two forms. The first form is the active form. So in the active form, your immune system can see it and fight it and get rid of it. But when that active form is under attack, it hides. It changes into a dormant form and hides in your brain. And then once there, your immune system can't surveil it. It can't see it, it can't fight it. And then, as you get older, your immune system isn't as great. It can switch back to the active form and come get you. And so it's difficult to vaccinate against because it has two forms. So we thought, why not make a vaccine that can attack both forms of this parasite? So we picked 6 antigens, 3 antigens for the active form and 3 antigens for the dormant form. So I made those vaccines and then we vaccinated animals. And what we showed here is that when you have an unvaccinated animal with Toxoplasma gondii, everybody dies around, within a month. And with the vaccination, we had 100% survival. And the reason we looked for 175 days is that by day 30, you should start seeing some of the dormant forms switching back to active, and you should start seeing some mortality. So the fact that we didn't see this for such a long time meant we had the infection under control. So this is very exciting. And this is applicable to other diseases like malaria, because it's a cousin of Toxoplasma gondii. So this is important. We can deal with potentially help us deal with these new types, not new types, but existing orphaned parasites that we have no product for to treat. So going after complex diseases, great. Another thing that we're really interested in is pan vaccination. Can we simultaneously vaccinate people against multiple diseases at once and have it work? So for this, went back to my friend Chris Cooper and said that, hey, why don't we look at 3 different types of filoviruses? These are biologically distinct. Zaire Ebola virus, and then we have Sudan Ebola virus and Marburg virus. So they're all filoviruses, but they are biologically distinct, different viruses. So we made what we call a multiplex vaccine, a vaccine that targeted an antigen on Zaire, an antigen on Sudan, and an antigen on Marburg, all in one. And one of the pushbacks we got was that if you make one of these multiplex vaccines, it won't work well. It'll compromise, and then you might not get a great level of protection against the other things. So it's better to kind of vaccinate separately. And the reason we kind of went after this is that we heard a lot of people were talking about alternative vaccination schedules, where they said, oh, don't vaccinate so quickly, space out all your vaccinations. And, that doesn't really make sense, but I think the best way to show that is with data, and there's data out there and we want to contribute to that data. So here's what we did. We went back and did an Ebola trial. So we made vaccines and then we give animals Ebola and see what happened. So here's what happens. We gave animals Zaire Ebola. Zaire Ebola, you should have 10% survival. And we see animals without a vaccine, again, it's 10% survival, that's normal. Then we made a vaccine against Sudan Ebola and vaccinated it and exposed them to Zaire Ebola. So a mismatch. They're not the same. And once again, only 10% survival because it's a Sudan Ebola vaccine and it doesn't protect you against Zaire Ebola. Now, we made a Zaire Ebola vaccine and we got 80% protection against Zaire Ebola virus. Great. But look what happens when we have multiplex. When we have all three in one vaccine, we maintain a very high level of protection. So it's great. So they're not interfering with each other. They're not doing anything. And it makes sense biologically. From an immunology point of view, you're always being exposed to a ton of pathogens all the time. And your immune system can handle it. I mean, the amount of immune system cells your body's making a day is staggering. So you're absolutely able to handle this sort of thing. So it was kind of great to see that. But with this, it's kind of really nice to say that, you know, mRNA vaccines are fantastic, but we also have to acknowledge some of the current limitations. And those limitations are antigens are made and processed by your mammalian cells. So when you use mRNA vaccines, you're sending an instruction to make a protein, make an antigen, and showing that antigen to the immune system and saying, develop immunity on this. If you ever see this antigen, it's connected to something deadly, destroy it. So that works really well for viruses because when a virus infects you, it hijacks your cell to make its proteins, to make itself. So viral proteins can be made by our mammalian cells. But for things that are very different from us biologically, like bacteria or fungi or multicellular parasites, they don't hijack your cell. They just get inside you and start eating you. So we don't have the cellular machinery to make antigens for bacteria, fungi, and multicellular parasites. So in this case, you know, you definitely want to embrace other types of vaccine technologies because the mRNA technology isn't a silver bullet. It is part of our arsenal, the ecosystem of protection that we can create. And it's important to recognize some of those limitations. But when it comes to viruses, mRNA vaccines are outstanding. But, you know, we're working on that. Even some of our work show that we can go after certain parasites like Toxoplasma gondii. So it is possible with clever design, but for now, something to remember and consider. So still early days for other types of pathogens, but for viruses, definitely we have that capability. All right. So one of the things that we look at is not just making super effective vaccines, but we're also really aware that our vaccines have to work for diverse populations. And this is why we often hear about, you know, embracing diversity and accessibility. This is what it means from an engineering point of view. Everybody is different. So we can all have DNA in us that encodes the same protein, but those proteins, the protein that you make might be very minutely different from the protein I make. And that's what helps make us unique. So if you look at a gene, genes are encoded by nucleic acids like DNA. And a single nucleotide polymorphism, so one nucleotide difference between person 1 and person 2, can really change how a protein works. And if one of those proteins are built into your immune system will work slightly different from the way my immune system works. And this is part of the reason why we have heterogeneous responses to things like vaccines. And, you know, some people get sicker than other people. Like, there are multiple reasons for it, but this is one of them. Genetically, we are slightly different, and this contributes to the heterogeneity of, you know, medicine's efficacy. So these single type nucleotide polymorphisms, you can use those to broadly categorize the human population. And we call those haplotypes. So if you're of one haplotype, that means that collection, that part of the population, all has the same single nucleotide polymorphism versus another haplotype who has a different one. And this is why it's important for your immune system. When you look at how a vaccine works, you have your cell, it got an mRNA or it got an antigen. In any case, there was an antigen inside, the antigen against which we want to develop an immune response. So that antigen, once it's inside the cell, this is what happens. Your cell degrades it into small fragments called peptides. It loads it on to a molecule called MHC class 1, major histocompatibility complex 1, and puts it on the outside of the cell. And this is how your cells show antigens to the immune system. So this shows it to the immune system, and then the immune system reacts to it and helps develop your immune response. So if you and me have different MHC class 1s, that can mean we have differing abilities to present antigens to the immune system. And here's an example. So this work is what we did in mice, and these are two different haplotypes of mice. So this mouse had an MHC class 1 that was different than this mouse, okay? So when presenting our antigen fragment, this mouse did a very poor job. So this is a negative control, this is a positive control. What we want to see here is a high bar. The high bar means you're doing a really great job of presenting the antigen to the immune system. A very low bar means you're doing a very poor job of presenting the antigen to the immune system. So you can see that this mouse here from this haplotype is doing a very poor job of presenting that antigen fragment to the immune system. So they're not going to really get a great immune response. This mouse, in contrast, is doing a phenomenal job. It's right up there with the positive control. So it's taking that antigen and really showing it to the immune system and doing it very efficiently. And this is the kind of things that we think about. We think about the antigen we're choosing, and we're thinking about, can they be loaded equally well across everybody, all populations, so that we can have equity in our protection. And this is an important thing that we are considering. This is something that my lab is very interested in, and because we want everybody to benefit from these wonderful technologies. So something to think about. The field is going here, and we really have to consider this because honestly, when you are thinking about a global pandemic, you absolutely want everyone to have equal measures of really great protection. So let's see. We have other opportunities too. So we talked a lot about how nucleic acids can be used in vaccines, but that's not the end of it. There's so many other applications that you can do, and I'll tell you about some of the ones that we're working on. Now, many diseases can have a variety of symptoms, but one of the most common elements that is shared across multiple diseases is inflamed blood vessels. So blood vessels, when they are sick or diseased, that is a common hallmark of a variety of diseases. So when you kind of take an engineering approach, we're always looking for new ways to address problems. So one of the ways in which we thought about this is that if you have the ability to make sick blood vessels healthy again, you could potentially help a lot of people, and for a broad variety of different diseases, whether it's arthritis or chronic inflammation or even chronic infections. These can potentially really help people out. So blood vessels are fascinating, and there is definitely an opportunity to look at this new opportunity to try to treat diseases. So to do this, we wanted to look at the RNA payload called the small interfering RNA, the siRNA. This is a type of RNA that if it gets inside your cell, the cell will use that instruction set to find a corresponding mRNA in your body and degrade it. So what this means is that if there is a gene that I want to temporarily turn off, I put in an siRNA against that gene, put it inside a cell, the cell reads it and finds the corresponding mRNA in your cells and degrades it. So that gene does not get expressed. So we turn off gene expression, and it's a temporary thing, so it's not permanent, so we can control it. So if you're sick, we can use this tool to shut down a gene that's causing you like a lot of medical issues. Now, we wanted to take it one step further. Now, we knew that I had a big interest in the bone marrow. So, and I'll tell you why. So I was developing a bunch of different delivery systems to deliver this siRNA payload to the bone marrow. So this is negative control, and we can see that with a couple of these materials, we were able to deliver our payload to the bone marrow. And here's why I was interested in the bone marrow. So the bone marrow is where a lot of your immune system cells are born, including monocytes. So monocytes are born in your bone marrow, and then they eventually leave, and then they go and help defend your body. Now, monocytes are great. You need them to heal from infection. You need them to heal from injury. They're very important cells, but sometimes there's a problem with them. When you have a heart attack, something bizarre happens that shouldn't really happen. When you get a heart attack, you get too many monocytes. So for some reason, you get way more monocytes in the heart than you need. So instead of the monocytes helping your heart repair itself, the monocytes cause a ton of extra inflammation, and they cause scarring. And when you have a scarred heart, it doesn't pump blood very well. And that's what causes people to have long-term morbidity when they have a heart attack. So we can see here, this is a cross-section of a heart for a mouse, and the red color is normal tissue, and the blue color is scar. So after a heart attack, you can see, look at this amount of scar on the heart tissue. It's not so great. So what we did is that we made nanoparticles that went to the bone marrow and temporarily shut the door so that the monocytes that were being made in the blood vessel, in the bone marrow, could not exit through the blood vessels, travel through the blood, get to the heart, and cause scarring. So when we shut the door, we got far less scarring. So this is a table quantifies it. So with the control, you see a ton of scarring, and then with our treatment, far less scarring. So this is an opportunity that you can really look at. Looking at problems from a very sideways approach is important. Not doing things the same as what people normally do. Let's find creative solutions, look at new entry points. And this is part of the fun of using this incredible tool that evolution gave us, nucleic acids, to kind of figure out what's going on next. So another thing that we're working on is, again, accessibility. And this is a life expectancy chart, and you can see that the dark blue is where people live the most, and in the lighter colors is where people have lower life expectancy. And this tends to correlate with poor living conditions and poor access to healthcare. And a lot of these countries are in the equatorial zone, so they're warm. They are hot countries, and they have limited infrastructure, including healthcare. So one of the problems that we have with nanoparticles and RNA nanoparticles is that they need to be stored at a cold temperature. So if you really want to make the lives of people in this region here better, you're really going to have to think about a way to give them nanoparticles that don't need to be stored in a freezer, don't need to be stored in a refrigerator, that can just work, because they need to be transported in the heat, they need to be stored in the heat, and they need to be administered in the heat. So this is how we can start looking at making this brand new technology accessible for everyone. So this is what another thing in our lab that we're really focused on, making nanoparticles that don't need to be refrigerated. So this is a graph that shows, in this graph, we are showing an RNA nanoparticle that's meant to silence a gene. So high bar means that the gene is active, and a small bar means the gene has been silenced. So what we did is that we took our nanoparticles and we made them, and then we treated them to cells, and we showed that they're working. And then we stored them for 3 months at room temperature, no storage or anything. And we also stored them in cold storage at 4 degrees. And what we showed is that there was no difference in performance. So we were able to make nanoparticles that didn't need refrigeration. In fact, when they were refrigerated and stored at room temperature, they had the exact same performance. And how did we do that? We designed a new RNA delivery molecule that didn't require cold storage because it was able to stabilize and keep the RNA safe and didn't fall apart, which is all the reasons why you need to keep it cold. So by being clever about your molecular design of your delivery system, you can make a big impact. And the last thing I want to talk about is that what else are we looking at doing? We're looking at beyond infectious disease. We're also looking at beyond heart attacks. We're looking at stuff like cancer too. New ways to fight cancer. So one of the things that makes cancer really hard to deal with is that they multiply really fast, but your immune system should be able to fight that. One of the reasons why your immune system has a big problem with cancer is that cancer cells are immunosuppressive. They are able to stop your immune system from fighting them. So this is called immunosuppression. They are able to send the right signals, recruit the right cells to protect themselves so that your immune system is unable to clear those tumors, because your immune system wants to kill those tumors. Those tumors are mutants. They have mutated DNA, and your immune system can see that and it wants to remove them because that can really cause you problems. But these tumors are able to recruit the right cells to protect themselves. So how are we looking at fighting that? Well, in some of my earlier work, what we found that a certain type of monocyte, so those monocytes are those cells, immune system cells that are born in your bone marrow, exit through the blood vessel and go to different sites of your body. We found that one monocyte, the Ly6C lows, seemed to be contributing and creating this immunosuppressive environment. So what we wanted to do was make a nanoparticle that would reduce the number of Ly6C low cells. And in doing so, we hope will diminish the tumor's ability to protect itself from the immune system, thereby allowing the immune system to come clear the tumor on its own. So we identified a gene, CSF1, that we felt would be great to reduce the number of these problematic monocytes. So what we did is that we made those nanoparticles, we injected it into animals, and we looked at what happened. And what we saw is that we were able to successfully bring down the proportion of these Ly6C low monocytes. And our hope and goal is to eventually deploy this as a method to help harness your immune system to fight off tumors by removing one of the key players that tumors use to protect itself. So by removing this, we're hoping to get rid of that immunosuppressive environment, or at least bring it down a little, so that the rest of your immune system can then come in and take care of that problem. So with that, I'm at time, and I would like to thank you all, but I have to acknowledge the phenomenal team that I have. So we have Anson, Julian, Alana, Grayson, and Janice. We have a few more incoming this summer and this September as well, but my blue coats. So they're called blue coats because we do a lot of engineering and chemistry, and we have to wear fireproof lab coats because some of our chemistry is a little pyrogenic. So it's a fun group. We have a lot of fun and we're passionate about the work we do. And of course, I have to thank all of our sponsors who have been helping us do this work: Medicine by Design, the Canada First Research Excellence Fund, NSERC, and the Canada Research Share Program. And of course, my collaborators like Chris Cooper, Bob Langer, one of the co-founders of Moderna. I was at MIT with him for a while. He helped me learn and do a lot of this stuff, kind of help shape the scientist I later became. Dan Anderson as well. Also at MIT, phenomenal person who works in nucleic acid delivery. So with that, my job now is to kind of pay what they gave me, the incredible training forward. So helping people like these become, their best selves and do things that I couldn't imagine would be possible. So with that, thank you so much for your attention, and I'd be happy to start talking about questions.
Thank you so much, Professor Khan. This has been a fabulous lecture filled with interesting details about your exciting research. It seems you have a really great team. I'm especially fascinated by the progress of development of vaccines from where science started 8 years ago and how the mRNA nanotechnology is allowing for development of vaccines at record time. Also importantly, as you have mentioned, in helping to develop vaccines for diverse population, and all the other exciting applications that could be developed for other diseases as well. We will jump directly into the questions from the audience. And we welcome your baby.
Sorry, he's sorry, he'll be quiet.
That's good. There is just a comment here. Somebody is saying, this is so fantastic. Fascinating. I need another lifetime to study this. I echo the same sentiment as well. This has been really great. One of the questions that came in, somebody is saying, as I understand, their claim, anti-vaxxers claim that mRNA type vaccines are new, untested, and risky. Given your statement that mRNA technology has been around for 10 plus years, is the anti-vaxxer claim simply incorrect?
Yes, it's incorrect, and here's the reason why. The only thing that the mRNA does is that it engages your immune system. But I think what a lot of the disconnect happens is it's just fundamental immunology you'll learn in for your first immunology course. It's that all of the cells that you use for protection are pre-made in your body. So your body randomly generates your protective cells. So it randomly generates the cells that make antibodies, and it randomly generates the T cells that help clear infection. So it randomly generates them. And then it holds on to them. It waits for them, those cells wait in your body, and then when they see a pathogen that they recognize, they attack it. So they're randomly made, and they're randomly made to recognize something. They are screened to see if they are autoreactive, and if those randomly generated immune cells attack your own cells, they are immediately eliminated. So they're eliminated, so the only immune system cells you have left are ones that recognize foreign material. And when they recognize foreign material, that means you're safe. They're not causing autoimmunity. So when you're exposed to a pathogen, that pathogen is shown to those immune system cells. And if one of those randomly generated immune system cells recognizes the pathogen, it takes action. So a vaccine doesn't create autoimmunity. A vaccine doesn't do any of this stuff. In fact, all it does is that it's just taking a shortcut to show parts of the pathogen to your randomly pre-generated immune system cells to say that, are any of you capable of recognizing it? If you are, come help out. And that's it. So it's something that I feel you can easily learn in high school if there was this opportunity to teach basic immunology there. And it's not a hard concept to grasp. So I think part of that is our challenge going forward for us as a society to think about sharing this basic type of knowledge with people when they're younger, because it's not hard to get. And I think with more general knowledge, people will be very comfortable with this sort of thing because it's not doing anything radical at all.
Absolutely. I really like it that putting in the emphasis on science for education for younger generation, probably it will help, even though it didn't help within this pandemic, but we hope for the best. So our next question is from Raymond Viola. If two vaccines are administered close together, one with an adjuvant, example, an influenza vaccine, and one without an adjuvant, example, COVID, could the first diminish the effectiveness of the second because of the presence of the adjuvant in the first vaccine?
Well, what we would do in that case, if you needed to get both done at the same time, is you do split dosing. If you really had to vaccinate at the exact same time, you would just inject one in one area and one in the other area, because it's a very local response. So the adjuvanting effect in our work showed that it was a very localized response. So if you are getting irritation at the site of injection, then that's where you get part of the challenge. So as long as that adjuvanting effect is happening somewhere else and not at where the mRNA is, your mRNA will have a much better chance of getting utilized by your cell.
Okay. And the next question is, how close is science in developing a vaccine for HIV?
So HIV is very challenging because of its rate of mutation. So it goes pretty quick. So when you kind of pick an antigen, it can mutate pretty quickly, and it also infects your immune system cells. So that kind of creates a bit of a challenge, because the cells that you need to fight are also actively being infected. So it's a very bizarre pathogen, and it's difficult to deal with. But we have many people who are working on it. In fact, my friend Chris Cooper, he's moved on from the U.S. Army, and now he's working with IAVI. So IAVI is a global nonprofit that works exclusively on making HIV vaccines, and he's doing the work, he's putting it in. He and other people, we're on it. We're trying.
That's very helpful to know. Another question from Miriam Jarsky. Do the delivery systems for these various methods pose a challenge?
And when you say pose a challenge, do you mean like in what context? Like, because they are in terms of manufacturing, it's less of a challenge, because you can make these delivery molecules ahead of time and store them, stockpile them. And then when you need to make a vaccine, you take a bit of that you made, and then you take a bit of the RNA, you put it together, and you're good to go. So it's pretty flexible. Safety is always the big one. Is it safe? Does it cause problems? And that's what the clinical trials are for. We actually go through a tremendous gauntlet of safety trials before you even get to a human, to show that these aren't causing any kind of problems. So only the ones that are absolutely safe and have been shown to be safe in multiple orders of animals, so not just mice, but higher order up, then can they be potentially even thought of to be used in a human. So it goes through a very comprehensive pre-screen.
Okay. And we have one more question that says, do you think that one's immune system is affected by one's mood?
Well, that is interesting. Okay. Immunologically, your mood can be affected by certain pathogens. So I'll spin it around. So for example, Toxoplasma gondii, when it's in your brain doing weird things, it can actually cause that inflammation and other kind of cytokines and stuff that happen that affects your mood. So it is conceivable that whatever causes those changes can impact what's happening to you. So whether or not what we're concerned is your medical condition causing some kind of systemic response, or is it caused by some systemic response? Because then that can definitely impact your overall biochemistry and hurt you. But in general, when it comes to your immune system has evolved over millions of years. It is phenomenal, and it works on autopilot. So in general, people's immune systems are going to be very robust. Otherwise, we wouldn't be here today, right? So you can trust in your immune system. It's going to be able to help you. There are other kind of autoimmune conditions that can cause flare-ups and stuff like that. And so there are things that, I don't have the answer to that totally, but I can definitely tell you that when it comes to infectious disease protection, your immune system is going to do a great job, because it's kind of evolved to do it on cruise control.
hopefully that answers the question and gives us a little bit more hope. The next question is from Akbar Nafa. So far, there seems to be a challenge with the lasting effectiveness of the mRNA vaccines. If we take Moderna and Pfizer as evidence, are there any success that the effect of these mRNAs will last long? How far are we from achieving that, or it always be to administer boosters?
So that is a very common misconception that we should debunk right now. All of the vaccines work very well, and they do give you long-lasting protective immunity, period. We have clinical data that definitively shows that that vaccine you got is still working now. So it's giving you long-term permanent immunological memory. The reason why they're not working now is because the pathogens evolved. So the antibodies, for example, that you've made are sticking to the original version of the coronavirus. As the coronavirus evolves, that same antibody, it doesn't stick as well to the new version of the virus. So that's on us, because we've let case numbers stay so high that we've enabled virus evolution. The best way to evolve a virus is to infect people. We are engines for viral evolution. And so that's part of the goal of bringing down infections, because that brings down viral replication events. And replication events are where the mistakes happen and the mutations happen. So you've made antibodies, they're great, and they can recognize coronavirus, no problem. But then you have a different version of the coronavirus, they don't quite recognize it. And here's what happens. When it doesn't quite recognize it, your immune system doesn't think to use that less efficient antibody against this new pathogen. So that's a problem, because your immune system then wants to restart the whole process. So it wants to restart your immune system process and make brand new antibodies. So it doesn't think that it can use the older, less specific antibodies to fight the new version of the coronavirus. Now, the way we've been addressing that is that we artificially induce what we call recall. So normally, you're vaccinated, you're good, and you get re-exposed. So you're vaccinated, you have high antibody levels in your blood, great. Normal, over time, your antibody levels reduce. They always wane, that is completely normal. Your body is not designed to keep antibody levels up forever. It reduces them. So you have a baseline small amount in your blood all the time. When you're re-exposed to the pathogen, your body then mass produces your antibodies again. That's called recall, and it comes back up, and then you fight off the pathogen and get rid of it. When the pathogen is mutated and it doesn't look the same anymore, your body doesn't recognize it, so it doesn't think to induce recall and bring up its old antibodies, because it doesn't quite match. It's like, it sort of looks new. I'm just going to restart my immune process. I'm going to start making new antibodies against this. And that's the problem. The booster lets us artificially induce recall. So now the booster brings back up those original antibodies we made. And yes, they don't stick as well to the coronavirus, but there are enough of them that we can clear the infection. So they're slightly less effective, but there are so many more of them that we overwhelm it. So that's why we have two jobs. Two jobs are bring down viral replication events globally, because then they'll slow down viral evolution. That's number one. And number two, we'll do our best to, if we can, slow down viral evolution, make updated versions of the vaccine as quickly as we can. And that's what we're doing. And also just to keep our vaccines as effective longer by addressing this problem as a global health issue. Because we can't expect stability here locally if we solve coronavirus infections here, bring down our case numbers, but in the rest of the world, it's skyrocketing, causing mutations. It's going to come back and get us. So we have to level the playing field, help everybody out, and then we all get out of it at the same time.
That sounds excellent. So probably we will end up with a high school student joining in here. And the question is saying, what advice do you have for a high school student who's uninspiring to be a scientist?
Yeah, I think the best thing to do is, you know, do the work, just study. And what I found is the most important is find out what kind of science you like the most. And some people think that this is what they like to do, right? They think that, okay, I like this kind of science, I'm going to do it forever. And you kind of get into it and you realize this isn't quite a good fit. But what's important in science is to kind of expose yourself to as many different fields in science as possible so that you can find what you're passionate about and what you love to do, because you're going to make a bigger impact when you love what you do. So my biggest advice is try as much science as possible, find out what's the right fit for you, and follow that, I think. And the other thing is that it's okay if what you thought was your original calling doesn't turn out to be your calling. It's okay. Everybody can discover that about themselves and try something different. So that's the most important thing. The second most important thing, I think, is to be honest with yourself. If you're not loving it, or if you think that it's hard, but I can overcome this, great. If you think that it's hard, I can't overcome it, but I'm not happy, maybe I want to try this other field of science, you can do that. So keep an open mind, explore as much as you can, and then find that fit.
That's a great advice for high school students, as well as for everybody who's interested in science in general, even at university level as well. Thanks everyone for the great questions. It seems the evening could carry on with more discussion, but we will have to end here. For final remarks, here is Erin Thompson, our fantastic president of the Woodsworth College Alumni Association.
Great. Yeah, as Huda said, it seems the evening could carry on with more discussion, but it is the end of the night. So on behalf of the Wordsworth College Alumni Association, Professor Khan, we would like to express our utmost and sincere gratitude to you for this insightful lecture. You made us all intrigued, and we come out of this lecture wanting to find out even more. We continue the tradition of the Alumni Cafe series for inviting lecturers with topics of interest, so our alumni community continues engaging and learning. This certainly has been a very educational evening. And again, thanks to everyone for joining, and thank you, Professor Khan. But before we say goodbye, we have two announcements. The first one is that we will be sending a short survey in the coming days, and it would be very helpful if you could take a minute to fill it out. We would love to hear from you. Our second announcement is that our next Alumni Cafe event is coming up April 26th, and it's entitled Web3 and the Implications for the Canadian Economy by Professor Don Tapscott, who is one of the world's leading authorities on the impact of technology on business and society. Please check out the details on the Wordsworth College events website, and keep an eye on your email for the invite. So that concludes our evening. Thank you again, Professor Khan, and thank you for everyone for attending. Thanks, and good night, everybody. Take care. Bye.